Hi all. So in this video we're going to see about cerebral circulation. So the previous university questions that have been asked in relation to this topic are special features of cerebral circulation, Monroe-Kelly doctrine and regulation of cerebral blood flow. So we'll see each one by one. First, we'll see about the special features of cerebral circulation. So when such a question is asked, we first have to write about the basic blood supply of the brain, right? What is the cerebral circulation? So we know that the basic blood supply, that is arterial supply of the cerebrum or of the brain, is mainly via the two carotid and the two vertebral arteries, which in turn forms a circle of Willis. So what is the circle of Willis? Remember, in circle of Willis, you might have studied it already in anatomy. We've got these internal carotid arteries, which forms the anterior circulation. So internal carotid artery will in turn divide to form the anterior cerebral artery and the middle cerebral artery. And we've got a communicating artery between these two anterior cerebral arteries, which are called the anterior communicating artery, right? And we've got another branch of this of the same, which is called the posterior communicating artery. Now the posterior communicating artery in turn joins with the posterior circulation. Which is the posterior circulation? It is formed by the union of vertebral arteries, which forms a basilar artery. The basilar artery in turn forms the branch posterior cerebral artery. So this is called the circle of Willis. So this is the main supply of the brain. What about venous supply? The venous drainage occurs via the deep veins and dural sinuses, and this in turn is going to drain into the internal jugular vein. So this is the rough introduction about the blood supply of the brain. Now what are the special features of cerebral circulation? So it is it is easier to remember by seeing this image. See, first you have to tell about the weight of the brain, or how much percentage of the body weight is the brain. Then talk about the cardiac output, talk about the oxygen consumption of the brain, and talk about the glucose consumption. So these, your answer should include these important 4 points regarding the weight, cardiac output, oxygen consumption and glucose consumption. And finally we'll tell about the Monroe-Kelly doctrine also. So we'll see each one by one. So see, the adult brain weighs around 1400 grams, which is 2% of the body weight. And of this, 60% is composed of white matter and 40% is composed of gray matter. So the brain consists of only 2% of our body weight. But what about the blood flow? The total cerebral blood flow is around 750 ml per minute, which is about 14% of the cardiac output. And this accounts for about 54 ml of blood per 100 gram of tissue per minute. So 54 ml of blood per 100 gram per minute and 750 ml per minute. Okay, so these values are important. And what, why are we talking so much about these the blood flow and all? Because if the blood flow to the brain decreases, it can cause problems with the consciousness levels, right? So the critical blood flow, if the critical blood flow is below 18 ml per minute per 100 gram, this can cause unconsciousness. So that is a feature of the importance of cerebral blood flow. What was the next point? It was about oxygen. So the total oxygen consumption is around 45 ml per minute, which is nearly 20% of the total oxygen consumption of the body. And the cerebral oxygen consumption is around 3.3 ml per minute per 100 gram. Basically the difference is in the unit. So it is 45 ml per minute as a whole and 3.3 ml per minute per 100 gram. Now another difference is that the gray matter and white matter has got a differential oxygen consumption. So see, gray matter, because it mainly consists of cell bodies, the oxygen consumption is around 3 ml per minute. Whereas for white matter, it is much lesser because it contains only axons. So it's around 0.35 ml per minute. Now here again, why are we so much concerned about this oxygen consumption and all? The brain tissue is highly susceptible to oxygen lack. So if there's a sudden cessation of oxygen supply, it can lead to loss of consciousness within 10 seconds. And if the lack of oxygen is for more than 3 to 4 minutes, it can cause irreversible brain damage. That is why we are so much concerned about this oxygen supply. Now what was the fourth point? Regarding glucose, right? So glucose is the chief source of energy of the brain. That is why hypoglycemia also can lead to damage of brain tissue. So these are the four important specialties of cerebral circulation. Now, the last important feature is the Monroe-Kelly doctrine. What is Monroe-Kelly doctrine? See, basically, Monroe-Kelly doctrine is that, see, a cranium here contains the brain, which is around 1400 ml, and then it's got the blood volume and it's got the CSF volume, right? Now, all these three are incompressible because it is enclosed in a rigid cranium. So, in case the volume of one increases, then the corresponding volume of the other two should be decreased because all these are present inside a rigid cranium. If it is not so, the ICP or the intracranial tension can increase. So the Monroe-Kelly doctrine is that the volume of all these three, that is the blood brain parenchyma, the blood volume and the CSF, should be maintained at a at a constant level. So see, the brain cranium contains the brain, blood and the CSF, and all these three are incompressible because of the rigid cranium. Hence the contents of the cranial cavity remains constant, and this is known as Monroe-Kelly doctrine. So now moving on to the next important section, which is regulation of cerebral blood flow. This can be written under the following headings: myogenic autoregulation, neural regulation, metabolic regulation, and the role of intracranial pressure. So we'll see each one by one. What is myogenic autoregulation? See, the cerebral blood flow is auto-regulated extremely well between arterial pressure of 60 to 140 millimeters of mercury. So this can be explained by the help of this graph. So see, if you are plotting the mean arterial pressure in millimeters of mercury on the x-axis and the cerebral blood flow in ml per 100 gram per minute on the y-axis, you can see that as the mean arterial pressure increases, the cerebral blood flow also increases. So now imagine what is a normal arterial pressure. So suppose it is say around 120. So if this is around here, you can see that at 120, if the graph is going to be straight like this, the cerebral blood flow will be around say 75 and all, right? But we can see that even when the arterial pressure increases from 60 to around 140, the cerebral blood flow is maintained at a plateau of around 50 ml per 100 gram per minute. So see, here we've got a straight line. Instead of going like this, the cerebral blood flow is maintained at a constant level. So this is called myogenic autoregulation. So even though the mean arterial pressure varies from 60 to 140, the cerebral blood flow is maintained at a constant level up to this 140 millimeters of mercury. So this is called myogenic autoregulation. See, after that you can see that the cerebral blood flow also increases as the mean arterial pressure increase. Okay, so this is the normal autoregulatory range. So how does this myogenic autoregulation work? So see, whenever there's an increase in the arterial pressure, see, when there's increased BP, when there's increased arterial pressure, what happens? There'll be stretching of the cerebral peak capillary sphincter. So the capillaries will be stretched because more blood is reaching them, right? So this will lead to opening of mechanically gated calcium channels, and thus there will be calcium influx, which in turn lead to increased myogenic tone and thereby decreased blood flow. So whenever there is an increase in the arterial pressure, there is a stretch on the capillary wall. So because of this stretch, there is opening up of mechanically gated calcium channels. So when the channels are open, calcium will enter, which will increase there will be an increase in the myogenic tone and thereby cause a decreased blood flow. Increase in myogenic tone means there will be vasoconstriction. The diameter will be lesser because the tone has increased, which will lead to decreased blood flow. Okay, so that is how myogenic autoregulation works. Next is neural regulation. Why do you need a neural regulation now? See, we know that during we set myogenic autoregulations up to 140 millimeters of mercury, but there are certain conditions where our blood pressure can shoot up more than 140. For example, during strenuous exercise and all, you can have an increased BP. But the good thing is that even when we have strenuous exercise or other state of excessive circulatory anxiety, the sympathetic nervous system constricts the large and the intermediate sized brain arteries so that the pressure does not reach the smaller blood vessel. This also can be explained with the help of this diagram. Here we said that up to 140 or 150 it is auto-regulatory range. Now suppose there is an increased state of strenuous exercise or any other increased circulatory activity because of sympathetic stimulation, you can see that the cerebral blood flow will not increase too much if there is sympathetic system activation. Okay, so that you can compensate for that increased mean arterial pressure. So that is the role of sympathetic system in regulation of cerebral blood flow. So whenever there's an increase in strenuous activity, there will be sympathetic nerve stimulation which will constrict the large and the intermediate arteries so that the blood flow is maintained at a normal range. Okay, so now we have an important regulation which is called the metabolic regulation. What is meant by metabolic regulation? See, suppose there's an increased carbon dioxide or increased H plus concentration. Whenever there's an excess of CO2 or excess of H plus concentration, cerebral blood flow also increases. How? How is that possible? See, whenever there's an increase in carbon dioxide, it combines with water to form carbonic acid. And we know carbonic acid will dissociate to form H plus and bicarbonate. This H plus can cause vasodilation of the cerebral vessels. So that whenever there is vasodilation, there will be increased blood flow. So this increased blood flow will carry these substances away from the brain tissue so that it can reduce the H plus concentration back to normal. So whenever there is increased carbon dioxide, there is going to be increased H plus, which will cause vasodilation of the cerebral vessels, which in turn will cause all these unwanted things to flow away from the brain tissue. Okay, so this also we can explain with the help of this graph. So to draw this graph, you can draw the arterial PCO2 in the x-axis, and on the y-axis you can draw the cerebral blood flow times normal. How many times your cerebral blood flow is going to change. So here you can see that the graph is somewhat like this, and the normal PCO2 is around 40. So you can see that as the arterial PCO2 increases, cerebral blood flow also increases, maybe up to 2 times. Okay, so this is how the PCO2 can act on the cerebral blood flow. Now another factor that is involved in metabolic regulation is oxygen. So oxygen deficiency is also a regulator of cerebral blood flow. So we know that our cerebral brain tissue, the brain tissue cannot survive without oxygen. So whenever there is a deficiency of oxygen, it ensures that there is a regulatory mechanism so that the cerebral blood flow remains constant. Okay, so what is the normal oxygen utilization? See, oxygen utilization by brain tissue remains within a normal limits of around 3.5 ml oxygen per 100 gram per minute, and the normal PO2 is around 35 to 40 millimeters of mercury. So what will happen if this PO2 is less? See, whenever there's a decrease in partial pressure of oxygen, what happens is it will cause vasodilation so that there can be increase in the cerebral blood flow. So whenever there's a decrease in the cerebral tissue partial pressure of oxygen below 30, it can cause vasodilation, which will in turn increase the cerebral blood flow so that more oxygen can enter the brain tissue and thereby compensate for this oxygen deficiency. So that was the second point in metabolic regulation. Next, a very important point is a local neuronal activity. See, there are 2 points in this. One is the role of astrocytes and other is the local vasodilator metabolites. What is the role of astrocytes? We know that astrocytes are cells that are present inside the brain tissue, and we also know that these astrocytes actually overlap or envelop the cerebral capillaries. So these astrocytes release various chemical substances including nitric oxide, arachidonic acid metabolites and adenosine in response to neuronal activity, thereby causing vasodilation and thus match the blood flow to the neuronal activity level. So suppose we are utilizing or we are seeing something or we are utilizing our visual ability to concentrate on something. So what happens? The blood flow to the occipital area increases. So based on the neuronal activity, the blood flow to that area increases. And they say the major cause for such a differential blood flow during each type of activity is this local neuronal activity, especially that of astrocytes. So astrocytes, based on the neuronal activity, will release various chemical substances which cause vasodilation and thereby increase the blood flow. And also the local vasodilator metabolites like potassium, hydrogen and adenosine all can cause vasodilation. So that is the third point in metabolic regulation. So in metabolic regulation we've seen about the role of PCO2, the role of PO2 and local neuronal activity. Now the last point in the regulation of cerebral blood flow is intracranial pressure. The role of intracranial pressure. So we already said about the Monroe-Kelly doctrine that the cerebral contents are within a rigid cranium. Okay, so naturally, suppose this represents the cerebral contents, the brain, spinal cord and the spinal fluid. There is a pressure exerted by this contents of the cranium, and that is called the intracranial pressure. Now we've got the blood supply to the brain. So suppose this is a blood supply to the brain. We know this is the artery which break into form arterioles and capillaries and which join together to form venules and finally drain into the internal jugular vein. So there is a pressure exerted by the artery which is called the mean arterial pressure, and there is a pressure exerted by the vein which is called the mean venous pressure, the central venous pressure. Okay. So the normal intracranial pressure is around 5 to 13 millimeters of mercury. So based on the Monroe-Kelly doctrine, we know that if any of the contents increases, the intracranial pressure also increase, right? So the body or the brain has got a mechanism by which it tends to compensate for that increase of the content. So suppose the blood flows more, it will try to compensate by decreasing the volume of some other factor so that the total content can remain constant. So this is the main principle of the role of intracranial pressure in regulation of blood flow. So see, there is a term called cerebral perfusion pressure or CPP. Cerebral perfusion pressure means the pressure by which the tissues are perfused by the cerebral blood flow. So the formula for that is mean arterial pressure minus intracranial pressure. So there are certain conditions in which our mean arterial pressure can be elevated. So in such conditions, we will see how the intracranial pressure will help to regulate such scenarios. So suppose there is straining or when our body has gotten downward acceleration, there is an increase in the mean arterial pressure. So whenever there is an increase in mean arterial pressure, the intracranial pressure also rises. So that we want the cerebral perfusion pressure to be normal. So whenever there's an increase in the mean arterial pressure, intracranial pressure also rises. And when intracranial pressure rises, you can see that it will try to compress the cerebral vessels. Okay, so when the cerebral vessels are compressed, what happens? The blood flow decreases. So the cerebral perfusion pressure is returned back to normal. Now this is good because this will actually protect against rupture of blood vessels by supporting them. So because there is whenever there is an increased blood flow, there is a chance that our small arterioles or capillaries may get ruptured. But because there is an increase in the intracranial pressure along with that, as per Monroe-Kennedy doctrine, what happens? It will cause compression of these artery cerebral vessels so that the blood flow decreases and it will prevent rupture of these capillaries. So that is what happens when there's an increased mean arterial pressure. Now suppose there's an increase in the venous pressure. Whenever there's an increase in venous pressure also, as per Monroe-Kelly doctrine, there will be increased intracranial pressure, which again will cause compression of cerebral arteries, which will cause decreased blood flow. So that the constants of within this cranium remains normal. And finally, there is something called Cushing's reflex. What is Cushing's reflex? See, suppose there is a brain tumor. The brain tumor can cause compression of the cerebral vessels. And when there is a compression of cerebral vessels, there is decreased blood flow, and thus that it can cause hypoxia at the vasomotor center. Now, this hypoxia at the vasomotor center will trigger the CNS ischemic response, which will cause intense vasoconstriction, thereby increasing the BP and restoring the cerebral blood flow. So Cushing's reflex is a type of CNS ischemic response which is caused by brain tumors which compress the cerebral vessels and thereby decrease the blood flow to the vasomotor center. So that is about Cushing's reflex. So that will complete the regulation of cerebral blood flow, in which we have talked the major four headings, which are the myogenic autoregulation, neural regulation, metabolic regulation, and role of intracranial pressure. Now moving on to some applied aspects, the most important applied aspect as far as cerebral blood flow is concerned is stroke. So what is stroke? It's a syndrome of rapid onset of neurological deficit that is attributable to a focal vascular cause. It's a rapid onset of neurological deficit. The person can have rapid neurological deficit because there's a focal vascular cause. There's a problem with the blood supply. So it can be due to ischemic stroke, in which there is a blockage of the cerebral artery by thromboembolism, or a hemorrhagic stroke which is caused by bleeding directly into the brain tissue. So the main treatment for stroke is you can have intravascular thrombolysis if there is a thrombus inside. You can have endovascular mechanical thrombectomy to remove the thrombus, anticoagulation so that it does not occur again, antiplatelet therapy in case it is due to a bleeding or hemorrhagic stroke. You can have anticoagulation and antiplatelet therapy, and anti-excitotoxic drugs so that there won't be any seizure or any other abnormal electrical activity. So that completes our short note on cerebral blood flow, wherein we have talked about the special features and regulation. So I hope this concept is clear. Thank you.